Hello everyone. So the next we will be discussing about the anaphase. So let's get started with the first slide of introduction of mitosis first. So what is mitosis? Mitosis is the process that partitions the newly replicated chromosomes into two daughter cells. So anaphase is one of the stage of mitosis. So what are the stages of mitosis? There is a prophase, there is a metaphase, then there is a anaphase, and then after that it is a telophase. So in this our main focus is on the anaphase. So this is a diagrammatic representation in which all the phases of mitosis are being shown. Here the prophase, in which there is a centrosome, there is a chromosome, and there is a centriole which is being seen. But in the next metaphase, what happens is these chromosomes will align or assemble in a at the equator of the cell, and this is known as metaphase plate because they are aligning in the or they are assembled in a one line. So, and these are the spindle fibers. There is a formation of spindle fibers which starts. This is a centriole which has been seen there. Then what happens in anaphase is the chromosomes will be at the opposite poles in the cell, and this is the spindle fiber. And in telophase, this will form the two daughter cells. This is the last phase of the mitosis. So we will be discussing, our main focus here will be the discussion of anaphase. So if we talk about the metaphase first, that first the prophase, then metaphase, then anaphase. So just before the phase of anaphase, there is a metaphase. So each sister chromatid of a metaphase chromosome is attached to the kinetochore microtubules via its kinetochores. So they're attached to the kinetochores. The chromosomes are attached to the kinetochores. So the opposite ends of these kinetochore microtubule, they're associated with one spindle fiber or one spindle pole. But at the anaphase, what is happening is the kinetochore microtubules, they pull the two kinetochores towards the opposite poles. So this is a representation in which it is showing that the chromosomes have separated and they are moving towards the poles. So this is the diagrammatic representation of how anaphase chromosomes are aligned in the opposite directions. So what happens is there is sister chromatids, they do not separate because they're held together at the centromeres by the multi-protein complexes called cohesins. So they are being held together at the centromeres. So cohesins, they are associated with chromatin and S phase. S phase is the part of the interphase where the S phase is known as the synthesis phase, where the DNA synthesis takes place. So during anaphase, what happens is this is a protein, this is separase, which degrades the cohesins. So they degrade the cohesins, and before anaphase, a securin protein that keeps the separase in an inactive state. Just before the anaphase, there is a securin protein which keeps the separase in an inactive state. The destruction of the securin is mediated by the APC by C complexes. So MCDK, they activates the APC-C functions in coordination with two homologous mitotic co-activators CDC20 and CDH1. So in this process, the anaphase work, and there are protein complexes which are present, and how the coordination of these works with the homologous mitotic co-activators CD20 and CDH1. So after the separation, each chromatin is pulled towards the pole. So we have seen the figure that what we have seen is that there was a kinetochore, then there were mitotic spindles, there were spindle fibers, and there were chromosomes which were attached. So they were at the opposite poles. They were at the opposite poles, and these chromosomes moved by two independent and overlapping processes. So they were moving towards the independent processes. So the anaphase can be divided into two distinct phases. So anaphase is being divided into two distinct phases, that is anaphase A and anaphase B. So these are the two phases which we will be discussing. So what is happening in anaphase A? So anaphase A is the initial poleward movement of the chromosomes, which is accompanied by the shortening of the kinetochore microtubules. So this force is generated by microtubule depolymerization at the kinetochore, which results in the loss of tubulin subunits at the plus end as the kinetochore moves towards the pole. So the movement towards the spindle force, they are facilitated by the kinesin 7 and kinesin 13, which motors that are localized at the kinetochore. So these are the kinesin 7 and kinesin 13 motors. They are localized at the kinetochore. Then what is happening in anaphase B? Anaphase B depends on the motor protein at the poles, and that pulls the poles apart as well as on the motor proteins at the central spindle that pushes the poles apart. So the two poles, they move further apart. It happens due to the sliding of polar microtubules past one another and pulling forces exerted by the astral microtubules. So a plus N directed kinesin 5 motor protein cross-link adjacent overlapping anti-parallel polar microtubules. In this slide the microtubules past each other, thereby pushing the spindle poles apart. So this was about the anaphase. So here is a diagrammatic representation in which anaphase A and anaphase B are being shown, which makes it more clear. So what was happening is chromosomes are pulled towards the pole side. So the chromosomes were pulled towards the pole side. So what was happening? There is a shortening of kinetochore microtubule. So there is a shortening of my kinetochore microtubule generated at kinetochores, and they move daughter chromosome towards the spindle pole. So they are moving towards the spindle poles. So these are spindle fibers which are moving towards the opposite poles. So this was the anaphase 1. Then what about anaphase B? They poles move apart. So there are poles which moves apart. For example, a sliding force 1, this is 1, is generated between polar microtubules from the opposite poles to push them apart, a pulling force. So in the second case, these are the marked as 2. What is happening? They act directly on the poles to move them apart. So they act directly on the poles to move them apart. So there are some forces which moves them apart. So these microtubules growth at plus ends of the polar microtubule. So this was about the anaphase A and this was about the anaphase B. These are all the references from where I have taken